Hi everyone, let's go through a few questions of this molecular basis of inheritance. Look at the first question. Select the correct statement from the following. E. coli synthesize RNA from RNA. This is not correct statement. Bacteria and eukaryotes do not synthesize RNA from RNA. They prepare RNA from DNA only. There is transcription. I can say that RNA cannot undergo replication. DNA undergoes replication. There can't be any RNA replication. Okay. There are few RNA viruses. They have got the enzymes RNA dependent RNA polymerase. Just find out mechanism of replication in such viruses. Replication. Find out mechanism of replication of viroids. Did you study that in first year syllabus? Viroids are infectious RNA molecules. How do they replicate? Is there any reverse transcription? No, there is no reverse transcription in viroids. There is direct replication. That means RNA dependent RNA polymerase catalyzes the synthesis of RNA from RNA. There is no translation or protein synthesis. Hence, viroids are called infectious RNA molecules. Just find out replication of viroids and the type of enzyme involved in that type of replication. In any case, bacteria cannot prepare RNA molecules from RNA. Bacteria and eukaryotes. This is wrong statement. DNA resists changes by repair process. Search this sentence in your textbook. DNA can undergo mutation, but it is evolved to correct that mutation. I make mistakes, but I try to correct it. There is repair process. There are certain enzymes involved in that DNA repair process. Anyway, we are not going to study the mechanism of DNA repair process. This is correct statement. This is the answer. One chromatid contains only one gene. Do you believe? Imagine this is one chromatid. It will have so many genes. I am talking about euchromatin of this chromatid. How will I identify number of genes? It can't be only one. I search number of promoters. One promoter, another promoter. Every gene has got a starting segment called promoter, terminator. You can recognize this thing in the NCRT diagram. Transcription unit. So there are so many transcription units in this chromatid. It can't be only one gene. See this whole thing, this is DNA. Find out what would be the length of this DNA. Even that question was asked in 2028. Okay, this is wrong. Telomerase is associated with nucleate. Totally misleading. This is incorrect. This is the nucleide. Where is the telomere? Where is the telomerase? There is no telomere. There is no telomerase. Hence, bacterium can undergo binary fission indefinitely. But every eukaryote has got this type of linear DNA. There is endpoint telomere, telomerase enzyme. Find out the function of telomerase. Telomerase activity decreases during senescence, aging process. In cancerous cells, telomerase is more active because cancerous cells are immortal. Because cancerous cells can undergo division indefinitely. Mitotic division indefinitely. Normal eukaryotic cells have lifespan. They do not undergo mitosis indefinitely. After some time, there is reduced activity of telomerase. There is loss of telomere. there is aging, there is death. So as long as the telomere is active, telomerase is prominent, there can't be any aging or death. I just check the chromosome of zygote, chromosome of dividing zygote, telomerase is more active. Okay, there can't be any senescence or aging in that early embryonic stage. Yes, so this is wrong. I have given the answer already. That is the correct statement. Which one of the following has split gene arrangement? Search this thing in your textbook. Split gene. Do genes of eukaryotes have what? Introns, exons, introns, exons, introns, exons? Definitely yes. That's how the genes are arranged. There is splicing mechanism. I will discuss the mechanism of splicing in another class. Mechanism of processing of hnRNA. Then this cannot be equal because bacteria do not have any split gene arrangement. They got only exons, no introns. Ethnogens belong to the category of prokaryote only. Even nostoc belongs to kingdom Monera. Species of kingdom Monera. It means prokaryotes do not have any split gene arrangement. The answer has to be second one. Fungi, protists, plants and animals have got split gene arrangement because all these are eukaryotes. Then once again go through 2020 NEET paper. There are several questions of this type. This type, not this question. It's fully on the NCERT concept only. Select the correct events that occur during transcription. You have to check the options later when we go through A, B, C, D statements. Alright. The main enzyme is RNA dependent DNA polymerase. We are talking about transcription. We are talking about RNA synthesis. Then why do we take DNA polymerase? It should be RNA polymerase. DNA polymerase catalyzes synthesis of DNA. It comes in DNA replication. DNA dependent DNA polymerase. Here it can't be DNA polymerase, it is RNA polymerase. RNA polymerase is dependent on DNA. It's not RNA dependent. So DNA dependent RNA polymerase catalyzes transcription. This is not the correct name of the enzyme. Only a non-coding strand is transcribed. It means the other one, the coding strand, is not transcribed. Only the non-coding acts as a template. Yeah, I will also discuss this mechanism separately in another class. So this can be a correct event, right? Primer is required for the initiation of transcription. Do you believe? Have you heard of primer? Where does this appear? This is the starting segment which provides OH group in DNA replication, right? It is removed later by the enzyme because it's RNA molecule. It is synthesized by primase enzyme. Okay, so this is not the correct event during transcription. Discontinuous synthesis takes place in bacteria. Wrong. Here it is continuous only, whether it is bacteria or anything. Continuous process. Can't be discontinuous process. No Okazaki fragments. We come across this discontinuous process in DNA replication. Do you remember leading strand, lagging strand? So this can be a correct event in DNA replication process. And then we got only one statement. The answer is this. Okay, students. So go through the NCERT carefully and think of this kind of questions. Bye.